A synapse is defined as the termination of an axon upon the membrane of another nerve cell. It is at the synapse where neurochemical transmission is affected, and thus an impulse is transmitted from one nerve cell to another. This ensures then the functional activity of the nervous system and its related areas as affects other organs within the body. A synapse, by definition, is that connection between two nerve cells and cannot be construed to be the connection of a nerve cell terminal to a muscle fiber or a nerve terminal to a gland cell. These are referred to as neuroeffector systems and classically not synaptic structures. A more detailed understanding of the synapse is important to the verification of the activity of drugs upon the nervous system. It is this chemical medium of activity from one nerve cell to another that is interfered with and mimicked by various drugs that we use today, whether they be analgesics, tranquilizers, or whether they be antibiotics. We would like to point out now some of the characteristics of the synapse, and an understanding of the ultrastructural study leads us to a better visualization of physiologic processes. The first drawing indicates that the synapse itself is the area that is indicated in this particular region. One can see the nerve fiber, nerve terminal here in diagram. This is known as the presynaptic area, and the area below the terminal is known as the postsynaptic area. If we take the small square here and bring it into higher magnification, such as this, we see that we have the presynaptic area here up to this membrane and the postsynaptic area after the membrane. The presynaptic area, which is a continuation of the axon, and the axon itself has contained a great deal of structures not seen in a terminal. In the terminal here we see mitochondria, clear vesicles or synaptic vesicles. These are the ones that contain the neurotransmitter agent, whatever the chemical mediator is. The chemical mediator then is released from these vesicles and has its effect on the postsynaptic membrane in this area. Postsynaptic areas also contain mitochondria, endoplasmic reticulum, and other organelles. As one examines then the presynaptic part of a synapse, we find that this particular area contains a very active chemical zone. This particular zone must replenish itself, must be activated constantly so that it can remain functionally active. This is done by transport of materials down the axon at a very rapid rate and also at a very slow rate. So there are two forces which transport materials to the presynaptic area or to the terminal of the axon. As this axon flow continues then, one gets an idea of the dynamic processes that are necessary at a nerve terminal. The next picture then shows us what a terminal will look highly magnified. These are the synaptic vesicles here, the round or oval clear structures. This is the presynaptic membrane, which is really a membrane of the nerve cell. There is a synaptic gap at this point, which is approximately 200 angstroms in thickness, and there is usually a convolution of the postsynaptic membrane to increase its surface area for more effective action of the chemical transmitter on this membrane, so that when an impulse reaches a nerve terminal, the chemical transmitters are released into the synaptic junction. They affect the postsynaptic membrane, and this cell then receives the impulse which was carried by the previous cell. If we look at the next diagram toward an elaboration of what actually is present within a terminal, we see, as indicated before, that regardless of the type terminal we have, there are still mitochondria present. There are the remnants of the neurotubules and neurofilaments that were in the axon, and there are the synaptic vesicles. Now, in a synaptic structure in a synaptic contact. These may be of several kinds, and the kind that are important to know about are those that are called axosomatic, that is where an axon terminates on the cell body of a neuron. There are axodendritic. Those are the ones where the cell body terminates on the dendrite of a nerve cell, and there are axoaxonic, which have a particular type of activity that we'll discuss now. As we look at this particular drawing now, we can see that this is an axosomatic type connection. This is an axon, a terminal, and this would be the soma, or the body, of a neuron. This represents an axon terminating on a dendrite, or axodendritic, and this is an axon actually terminating on an axon. This is an axoaxonic synaptic structure. Each one of these configurations has its own effect and has its own type activity, and oftentimes different chemical agents are responsible for different responses. The total response of a synaptic transmission is that one nerve cell depolarizes the membrane of another. This is the way that impulses are transmitted. If we were now to look at other drawings to determine what kind of terminals might occur, for instance, here is two types of synapses on a dendrite that may have totally different configurations. This may be either due to different transmitter agents present or a different effect of the transmitter agent on the dendrite. This part of the neuron is receiving impulses that will be carried to the cell body, received from these two. It is possible that this particular axon could be excitatory to this membrane, and this one could be inhibitory, or the vice versa. At any rate, different terminals have different effects on the particular membrane which they supply. The next drawing gives us some idea about why we would want to investigate the synapse in such detail, because it's conceivable that one neuron can have as many as 100,000 synaptic contacts on its surface. Now, no neuron in your body or mine is large enough to accommodate this number of synaptic contacts. Therefore, the neurons have elaborated their membrane system into what we call spines or gemmules on the surfaces, so that no membrane is smooth on a neuron, but that many are developed into spines. Figure 4 then shows us that these little pointy surfaces, or these varied surfaces on these processes of this neuron, give extra areas for contact for other nerve cells. This is a special silver stain of a neuron, so that it's stained alone without other cells in the way. The synaptic contacts would be hundreds of contacts along each one of these type spines or gemmules. If we look at the next picture, we can see a high magnification of this. For instance, here is a dendrite, and this is a gemmule or a spine from a dendrite. Here is an axon terminal, so we have a synaptic junction. We find that this type activity is quite often seen in these areas. The next picture shows even a more elaborate configuration, and that is the spine of the dendrite itself. The spine has developed into many convolutions, and one synaptic structure from one axon is innervating this entire area. So the area of contact here is markedly increased, and the synaptic activity can be considerably varied and have pronounced effect on the dendrite. If we begin to consider then the types of transmitter agents that are responsible for synaptic activity, probably the first one that was discovered was acetylcholine many years ago. Within the time of the past 3 decades, however, other transmitter agents have been discovered. Some of these are norepinephrine, 5-hydroxytryptamine, gamma-aminobutyric acid, and now we're finding in the present day and time that other agents, polypeptides and hormones, are important in regulating the membrane activity of a neuron. Synapses then have definite activity and regulation of cell activity in relationship to these hormones. We can take a look at another set of drawings that will show us, for instance, the type of transmitter agent that is characteristic of a synapse. Noradrenergic simply means that the synapse uses the chemical compound norepinephrine to mediate the response. That is, norepinephrine would be released from this hypothetical terminal to depolarize the membrane of the adjacent cell. Norepinephrine is characteristically stored inside of a synapse in what we call granulated vesicles or dense core vesicles. This is very characteristic of a noradrenergic synapse. Other compounds related to norepinephrine, for instance dopamine, are stored in a very similar manner. We also have mitochondria present, which is a characteristic of all synapses. The next diagram shows us what a cholinergic synapse would look like, and that is a synapse that in which the acetylcholine is packaged inside the synaptic vesicles. Occasionally there is a dense core vesicle, but this is a different characteristic than those that are noradrenergic. Mitochondria, again, are always present. The characteristic feature of these terminals is that the synaptic vesicle is a membrane-bound structure, and the membrane itself is not too different from the membrane that surrounds the synapse, so that as the vesicles move toward the membrane, they fuse with the membrane, and their content is released into the synaptic gap. The next drawing shows us another type of synapse that may contain a different kind of substance, and it should be understood now that not all knowledge is available on exactly what is in a nerve terminal and what chemicals mediate synaptic activity. This could well be one that belongs to gamma-aminobutyric acid, or better known as GABA, or it could be a terminal that contains 5-hydroxytryptamine. These are some of the variabilities that affect us now, but by and large, we know that a transmitter agent is stored in a vesicle. An impulse travels down the axon to release a chemical from the vesicle. The chemical then affects the postsynaptic membrane. As we look at the next drawing then, we can see that some terminals do not contain any synaptic vesicles at all, only mitochondria. It has been hypothesized that this type synapse, probably which occurs in the inner ear and some parts of the taste buds and olfactory mucosa, may well be sensory in nature and receives impulses rather than sends impulses. The next drawing is gives a configuration of what actually happens at a synapse as to what we've discovered. I think it's important to point out at this time that a neurotransmitter agent is largely formed in the presynaptic area. The neurotransmitter agent generally is not transported down the axon. The materials for the formation of that transmitter agent are. The neurotransmitter agent is formed at the terminal and presynaptic area. The enzymes and the precursors for its formation are present, as are some degradative enzymes. As the transmitter agent is released into the synaptic cleft, receptor sites are available for it to act on its effector, which is usually another neuron, and there are also degradative enzymes at that point that destroy the transmitter so that we do not have constant depolarization of the postsynaptic membrane. You can see now that there are a number of areas, either precursors, enzymes for formation, releasing agents, agents in the synaptic cleft, receptor blockers, degradative enzyme blockers or activators that in many ways can affect the level of activity at a synapse. Pharmacological intervention in these areas has come to be extremely important and a useful tool in treating many different diseases and maladies of mankind. Now let's take a look at this particular drawing that's right here and get an idea of what the presynaptic terminal is. We have mitochondria present. This happens to be an adrenergic synapse. It will release either norepinephrine or dopamine. The synaptic vesicles are present here. In this case, tyrosine is brought in and converted by enzymatic processes to DOPA. DOPA then is converted by a specific enzyme to dopamine, which is referred to as DA here. DA then is converted to norepinephrine. And norepinephrine, by a process, is released into the synaptic cleft. Norepinephrine then will combine with a receptor on the postsynaptic membrane and depolarize, result in depolarization of this cell, of this terminal in particular. Then dopamine, or norepinephrine itself rather, will be degraded into non-active byproducts, or it can be taken back up into the presynaptic area. So in this case, we have two methods of relieving the synaptic cleft of its transmitter. It can either be degraded by the enzyme that destroys norepinephrine, or norepinephrine can be taken back up into the presynaptic area. In addition, there is some loss of norepinephrine, though minimal, as it attaches to the receptor. These are normal biologic processes that ensure functional activity and conservation of energy. This particular type activity is present in the nervous system, in the central nervous system, and in the autonomic nervous system. As one looks at other types of transmitters and their effects, we take a look at the next diagram, and we get an appreciation of what can really happen at a neuroeffector organ. We will follow through now a sequence of drawings to show a typical cholinergic response. This can either be on a nerve cell here or on a muscle cell. This is an axon with an action potential that's depolarizing this presynaptic membrane. Acetylcholine is stored in these small vessels in here. Here is a synaptic cleft. This is a postsynaptic membrane, and this is the postsynaptic cell. The action potential travels down the axon, depolarizes the terminal. Calcium and potassium influx and efflux are activated, and acetylcholine is poured out into the synaptic cleft. As the next drawing will show you then, acetylcholine itself will attach to receptor sites. As these receptor sites are attached to, they are affected. The entire polarization of this membrane is changed then. Sodium and potassium are flow in and out. The bioelectric potential of this membrane now, of the postsynaptic membrane, begins to change, as did that of the presynaptic previously, and we find out in the next series of diagrams that what happens is a potential is generated in this membrane so that we have an action potential, a total depolarization of the postsynaptic membrane, while the presynaptic terminal is restored and ready for another response. These pictures then summarize for us what happens at a synapse. As you can see, the important aspects, obviously, of synaptic activity are an intact nervous system, especially an intact neuron with its axon, the constituents of the axon, and its connection then by its bulbous termination, the terminal, the axon nerve terminal, and its relationship to the postsynaptic site. Keep in mind that the postsynaptic site can either be the cell body of another neuron, can be a dendrite, can be an effector organ, or may even be another axon. It's important to understand the chemical processes that occur, the enzymes that are necessary. This is one unique portion of the body through the high magnification, sometimes up to 500,000 times, of the electron microscope, that one can visualize organelles, small structures that are functionally related to the nervous activity and to the chemical mediation of nervous activity at the synapse.